Hello and welcome to Omero and Image Data Management. I'm Doug Cromie. Let's get started. In some labs, students come to the core facilities and they may collect images from the microscopes in the Leica format, the Nikon format, the Olympus format, the Zeiss format. Perhaps they'll put them on a flash drive or a portable hard drive. And back in the lab, there's a faculty member and a grad student and a postdoc. They have access to cloud storage, maybe an analysis computer, and there's other students in the lab, undergraduates and first-year grad students. And the data takes a somewhat tortuous path through all these different things. It may get converted to TIFF, maybe mailed back and forth. It's got multiple versions, goes in multiple locations. And while I can't say that I know that this has happened to other labs, I can't imagine that it hasn't, because sometimes bad stuff happens. You have students that maybe left the U of A and have their access to their Gmail account now is locked, and any data that's there is unavailable. Somebody in the lab's computer is going to die. It happens. Maybe the flash drive walks away or goes through the wash. And there's really a number of problems. There's multiple versions. They may be in multiple file formats. Any acquisition metadata that came from the microscope is possibly lost. And there really isn't a provenance that is this an original or modified piece of data. It's really difficult to locate the original data, and some data may get lost. And quite often, important experimental metadata is in non-digital formats like lab notebooks, hard copy lab notebooks. And it's a problem because there's just a lot of data. And if someone asked you to find something from a year or 2 or 3 ago, you'd be hard-pressed to find it. Now, the NIH and the NSF are requiring data management plans. The NSF has been at that for a couple of years now, and the NIH started a year ago in January 2023. They are requiring not only that you have a plan for managing, but also eventually for sharing. And unfortunately, sharing, like this cartoon shows, used to involve a whole lot of miscellaneous storage formats. We won't even talk about zip drives and jazz drives and magneto-optical drives and floppies and all those things that go back a ways. But many of us remember, oh, we don't have something that reads that anymore. So the U of A has begun offering a local instance of Omero, which is client-server software for image data management. We'll talk more about that as we go along. Now, for managing and curating information, the high-end instrumentation that you'll often find in a core facility will capture things about laser powers and filters and exposure times and those sort of things, magnification. But there's a lot more to reproducibility than just how the images were captured. What animal species is it? Or is there patient information? Or is it a cell culture line? What culture conditions? Was it done with serum-free medium, or did it have other indicators in there? What organs are those that are being looked at, or were they fixed? Were they flash frozen? Are there controls, treatments? What other conditions are there? Who did the experiments? Was it the best person in the lab or the cringeworthy person that you're really not sure whether their lab skills are up to the job? What were the staining conditions, the dilutions, the lot number of the dyes, the reagents that were used? Maybe some other experiments done in the mass spec lab or the genomics lab. And if you did any post-acquisition image analysis, what was your pipeline or your methodology for doing that analysis? There's a movement in science to move towards making data more findable, accessible, interoperable, and reusable. And this cartoon shows, people acquire data, but sometimes when they make it available, other people can use it and sometimes use different data sets and combine them and come up with new information while citing the source of the original data. A lot of big meta-analysis in clinical studies use these sort of things where they pull data from multiple studies and try and come up with in a sense, a larger pool. The image data resource is an example of well-curated image data that's available for reuse, and it's online, and others can use it, and as long as they properly cite the original author or lab that created the data. There's a number of problems with microscopy data. Microscopy data can be very complex. We have XY information, we may have Z-stacks or focal information, multiple fluorescence channels, or even spectral information, time information. And if we're doing a very large tile scan, we may have locational information. And all of that comes in a proprietary format because the vendors have different ways of storing and taking into account what data is important to know about for these different multiple dimensions. And it's rare that one vendor can open another vendor's files, so it leads to this proliferation of single-use software, which is rarely friendly to Mac users because very few scientific softwares run on the Macintosh OS. And the files can get very large. Slide scans can be 5 to 10 gigabytes or larger. If you do multi-channel slide scans, multi-channel fluorescence slide scans, they can be in excess of 100 gigabytes. And while we don't have anyone that is hardcore into light sheet imaging yet, raw data sets and light sheet imaging can sometimes be several terabytes in size. And there's things like high content screening as well, which can create a lot of data. So why Omero? Well, it's been under development for about 20 years, so there's been a lot of thought and a lot of work put into how to work with image data. It uses the bioformat plugin that's often part of ImageJ that comes also from the same group, and it allows us to open over 160 different file formats. There are estimated to be about 1,000 working Omero servers worldwide, and at least 3 countries that I'm aware of are making a really big centralized push to use Omero for data management and sharing. And the image data resource that I mentioned before is up to 385 terabytes, and that runs on Omero. The funding for Omero at the University of Arizona came from a larger project, the Health Sciences Strategic Plan 5.3, which is sometimes called the Analytics Powerhouse. And there's 3 parts that are worth noting. Soteria is a place for people that need to work with personally identifiable information or clinically relevant information that has patient information embedded in it. Within that, you can use REDCap, Open Specimen, which is a biorepository software. The tools from Cyverse have been brought into that so that they can be used, and they call it Cyverse Health. And there's also secure access to the high performance computing. So if you're working with clinical data, this is a resource, and really a unique one because it's one of the first in the country to be like this. Omero is, as we'll talk about, an image-centric database. That number 150 is the latest I've seen is now over 160. If you happen to be someone that's working with medical imaging type data like MRI, CT, ultrasound that comes in the DICOM format, there is also software called XNAT that's available for people doing research in that field. And just as a side note, while Soteria is secure for working with patient-related information, Omero and XNAC do require de-identified information. Omero is not another form of cloud storage. It really is intended for curating and managing bitmap data. It can do a lot more than just stick it in a folder and let it disappear. It works with microscopes and other instruments, even if it's in a proprietary file format. We have a little 100 terabytes of allocated space, but there's the ability to get more. We encourage our users to think about all the information that needs to be stored with that image data so that it is reusable. Because the joke is that the most likely user of this data is your lab. But 5 years from now, future you will try to get information from past you, and past you does not respond to emails. So it makes viewing and sharing data, as you'll see, much easier. Captures the image acquisition metadata, and then there has the ability to add structured metadata such as key value pairs, tags, regions of interest. And if you're one of those people that loves the starring system on Amazon, there's actually a way you can star your pictures so that if you come across one that's just stunningly beautiful that you'd really love to use in a publication, give it 5 stars. And some of these other aspects of a data management plan can be worked out with the Data Science Institute. So we can capture images, bitmapped images from digital cameras, core facility or lab microscopes, anything that captures a digital image, high content screening, gel readers, DICOM images. This Omero would be a place if you just did an occasional experiment that required you to have DICOM images. And really just about any device that creates bitmapped or pixel related data. Our instance of Omero is behind the campus VPN, so you do have to log into that to get access to it. And you can upload data either using a desktop client that runs on Windows, Macs, or Linux, or if you're a coder, you can write custom Python scripts. There's a lot that can be done with Omero using Python. I am not a coder, so you won't hear much about that from me today. And accessing the data really only requires a web browser and credentials to log in. It's stored in protected lab groups, so even if your lab and the lab next door are on Omero, you won't be able to see their data and vice versa unless you give them permission to do that. And the faculty members control the permissions for their groups. With Omero, you can organize your files, add additional metadata, search, view large and complex multidimensional data, connect to your data from an image analysis software programs. Many of them have plugins that will directly connect to Omero, allow you to perform an analysis, and then put the results of that analysis back up in a file that's attached to the image data. And you can create publication-ready figures that reference the original data, and most importantly, do not change that original data. We have a couple of projects going, and we're looking for more. Dr. Curiel, the Cancer Center, is working on a collection of images taken in the clinic of skin lesions, and they use just a regular digital SLR camera. They have clinical data that they've acquired in REDCap, and if there's a biopsy done, that information goes into open specimen. All that's connected through a unique identifier. And then with Python scripting that the Center for Biomedical Informatics and Biostatistics did for them, the images are converted from JPEG to DICOM. They're taken from the UA Box Health folder that they've been placed in. A query goes out to the REDCap data and actually sorts the images to put them in the appropriate folder based on a diagnosis. And this was featured a few months ago in Health Sciences News. Dr. Zavros is working on a tumor atlas for pituitary neuroendocrine tumors. She's also working on one for pancreatic cancer. And she wants to combine spatial transcriptomics data. If you're not familiar with that term, that's basically looking at DNA and RNA expression on a tissue section, along with highly multiplexed fluorescence labels of the slides. So what proteins are actually being expressed using up to 20 channels of fluorescence on a single slide. And then she wants to combine that and create an atlas of these tumors. And the de-identified image data has clinical data as well as biospecimen data. And we want to have an image analysis pipeline that can take these data-rich sources along with the clinical information and try and make sense of what's happening in these types of tumors. She's had several grant applications that have gone out and they've received good scores, and we're waiting to hear about funding. This project is just getting started. This is 4 faculty members, Dr. Wilson, Goodrum, Thorne, and Paik from 3 different departments. And they're looking at viral infections and in the colon. And one of the things that's really beautiful about Omero is it's going to allow these 4 investigators who are in 4 different buildings, starting from the Cancer Center, Life Sciences North. I don't know if Dr. Goodrum's in MRB or in Bio 5, and then Life Sciences South, which is a good mile away from the Health Sciences Center. This is going to allow them to share their image data much easier than they have been able to do before. So let's take a quick demonstration of what Omero can do. I'm going to jump out of this slideshow for a moment and open my web browser. So here we have a scanned microscope slide. This is about 475 megabytes in size. We can zoom in. You can see it's refreshing the screen and filling in the information. This is what's called a pyramidal file format. If you've worked with Google Earth before, you should be familiar with this. You can see whatever on your screen is at high resolution, but it doesn't have to load all the data at high resolution. So you can move around. You can see down here that we have the location on the section. If I click to a different spot, it moves me there and then refreshes the screen. And I should say I am doing this on a couple-year-old laptop on Wi-Fi. So this is you know, if there's a little bit of a refresh lag, that's why. Let's look at something else here. So this is an image from a confocal microscope, and I'm not a big fan of fuchsia. Let's make this red. And I'm waiting for it to update. Oh, I turned it off, that's why. There we go. So here, I made that a little brighter so you can see it. And now I'm going to change my Z-plane to different locations, and you can see that it updates as it pulls in new data. This is a, just to show you that it doesn't have to be from a microscope. This is data from a gel reader. And if we adjust the histogram a little bit, we can see a little bit more detail in it. And just to prove that it isn't just that kind of data, when we were working with the skin biopsy group, just so that we weren't moving a lot of patient-related images around, I said, just send me some pictures from the camera. So they took pictures in the parking lot. So this is a JPEG image that we uploaded. So now I'm going to show you something that's kind of interesting. This is a very complex image that came from the Zeiss confocal on main campus. So I'm going to open this image data set. So you can see over on the right, this was using the spectral detector on the Zeiss there, and this is raw data, so it gives us the individual channels for the entire spectral range of that detector. Not all that interesting, but let's look at it unmixed. So this is the unmixed data. This is where they take and process it so that the autofluorescence is removed. So we can turn channels off. For example, I can turn the yellow off. This is actually pretty large data because it's multiple panels stitched together. And we can go to different focal planes and see different information. So here's an example of a scan slide in fluorescence. Now, I'm not real happy with how this looks, so I'm going to change some of these values to something I've figured out a little bit ahead of time. So that makes the blue look a little better. There's some more green. Actually, not a whole lot in the Psi 3, so I turned that off. Turn the Psi 5 on. Set it to 8,000. And so we have 3 of the 5 channels turned on. And we can zoom in. And you can see it's refreshing with new information. The beauty of this is I have changed the settings for how this is displayed, but I have not changed the underlying data at all. And that's something that's really kind of special about Omero, is that the raw data stays as raw data. And now there's one other thing that's kind of fun, is this is called Omero figure. And every image in Omero has an ID number. So I'm going to put in a number of images that I've already selected and have it create a figure for me. So if I go up here and I move that around, I go, gosh, I really need to rearrange that. That just put it back the way everything's nicely lined up. You know, I want to put a scale bar on here. And I would prefer my scale bar to be black there. And I'm going to make that 50 microns. There we go. Make this one 50. Make it black. This one I'll make 50, but I'll keep it white just for contrast's sake. Make this one 50. Now, I'm kind of interested in this image, and I want to look at it a little differently. I want to put this one piece nicely in the center, and I'm going to zoom in on it so that it's very much in the center. Get this single neuron. You see what happened here with the scale bar? That rescaled itself in real time. You can also do things with labels that include information from the key value pairs or other metadata, so that you can have cell lines, treatment groups, things like that, that show up as column and row headings and such as that. You can save this, and again, the underlying data is not changed. I didn't crop this image. I just zoomed it in for this particular function. And you can also export it so that you can go straight to your publication. Let's go back to our slideshow and wrap things up a little bit here. If you're interested in Omero, if what you've seen is interesting to you, send us an email at our project email. Encourage you, if you're thinking about Omero, to consider starting with a small project that's kind of manageable. That way you don't get overwhelmed by collecting all the metadata that you might not have realized you needed to do. We do have a pilot document that we ask people to complete so that there's a way for us to understand what you want to accomplish, and also so that we can all stay on task. We're here to help, and just a reminder that everything has to be de-identified. And we will ask you for feedback. Did this work well? Are there issues that we could do better? Right now it's free. If you want to learn more about the strategic plan, there's that link. We had a 2-day training workshop in August of 2023, and recordings of that workshop are available on D2L. And if you email us, I can give you access. And then there's some additional information on a post-workshop webpage. So I want to thank you. If you want to send us an email, you're welcome to scan this QR code with your cell phone, or you can just write down the Omero address, and we are happy to help. And I'm just going to put some links here for the images that I used, and thank you very much.